Thanks everybody for joining us for this ERI seminar. It's an absolute pleasure to reintroduce David Schwally to you guys. David is visiting us this week on his journey through New Zealand, connecting with researchers in a collaborative project. So hopefully we hear a little bit about this today. I'll let David introduce a little bit more about himself actually, but David and I met when you were on your sabbatical here as a Fulbright scholar in 2020 and months during COVID lockdown, and we just shared some stories around about that time earlier this morning. David and I connected through some of the lakes research that we're doing here and the drone work that David has been involved in terms of using drones to study lakes and collect samples from those lakes. It was a really exciting experience. David, you left a really lasting impression on the group, really, with your enthusiasm and positive attitude towards all these projects. And we're really grateful that you came back to visit us. And we can't wait to hear what you have been up to in the last few years. So thanks, David, for coming along.
I appreciate that. Thank you, Dennis. It's great to be back. I left New Zealand a little over 3 years ago, the 15th of June, 2021. And I'll tell you a little bit about the journey, my time in New Zealand, of course, the science that we've been doing both at Virginia Tech and then while we're here in New Zealand as well. So hopefully this will advance. I'll mention right off the bat, I'd like to throw the sort of acknowledgements at the beginning of my presentation. So this is my lab group, which is of course a big part of the operation back in Blacksburg, Virginia. We didn't get the colors quite right on the plugin, which is what we were trying to sort out, but those are different colors of little smoke emitters. I'll tell you a little bit about them later as part of a tracer that we're using to track plumes of things. We've been funded, of course, by the National Science Foundation, our prime sponsor in the United States Department of Agriculture, Fulbright, New Zealand, which I'll tell you more about today. We have a project funded by NASA looking to transport of African dust, ICTAS at Virginia Tech, and then a center which I now direct, the Translational Plant Sciences Center at VT. I'll start by introducing former Senator Fulbright from the great state of Arkansas. It's his program, his international exchange program, that actually brought me to New Zealand for what was supposed to be 6 months, but ended up being a year and a half. Former Senator Fulbright was, of course, quoted as saying, "The essence of intercultural education is the acquisition of empathy, the ability to see the world as others see it, and to allow the possibility that others may not see something we have failed to see." And certainly during the pandemic and during post-pandemic years, that statement is ever true. So I'll tell you a little bit about my journey first to New Zealand. We landed in Christchurch on the 1st of January, 2020. This is, of course, me at the center. My wife was taking a picture of my two daughters, Nora and Vivian. We spent the better part of the month on the South Island during what we thought at the time was going to be our only journey to the South Island. And then we landed, of course, in Hamilton, a little cottage, a grandmother's cottage in Tamahiri. My Fulbright formally started February 1st of 2020. This is, of course, Fulbright headquarters, Penelope Borland, who's still directing Fulbright, now Fulbright New Zealand based in Wellington. And then we quickly shuffled off to a marae in Wellington where we proceeded to spend the night. This was quite an experience for somebody like myself. And I was actually quite smartly in this bed on the end, which was close to the restroom. So all of the Fulbrighters, of course, were there and members of the community and of course Fulbright New Zealand, and we spent the better part of a day having a variety of correos right here, talking with members of the community, and of course this was a cultural immersion at its finest for us. And then settled into our block, which is now Whitney's office, of course, as she's enjoying. And I was back there in the corner, and of course all my equipment stacked up in here, shipped over all sorts of drones, among other things. And that was my home for what was only supposed to be 6 months, but turned into a year and a half. My two girls, of course, were going to school at the Tamahiri Model Country School and also at Berkeley. For those of you that have children or aware of those schools, and uniforms are a new thing for them. That's not something we're used to in the U.S. And so, of course, they're all get it out for that. March 2020, received a letter from then President Trump, said, "Hey, Fulbright program's been canceled worldwide, come home. If you don't come home, we may not be able to get you back." And so my Fulbright started the 1st of February and it ended March 9th with a certificate of completion and go home. And so we of course decided to stay. We figured out that, hey, being on this beautiful island nation maybe wouldn't be such a bad thing, particularly given that a science-forward agenda was being promoted by then what my kids called anti-Jacinda, versus what was happening in the U.S. Within President Trump. Of course, the 17th of March, be strong, but be kind, we will be okay. Words that, of course, many of you remember for those of you that were here. And of course, Ashley Bloomfield, this will require all efforts. Strongly urge all New Zealanders to play their part. And of course, that constant messaging, the team of 5 million, and we were glad to be part of that team of 5 million during the pandemic. Of course, you might recall coming out of that 7-week lockdown into this sort of phased level strategy, which was deployed and changed as cases sort of broke through the MIQ system, but ultimately allowing us to reemerge and do some rather exciting work. Here's Brendan, I guess, making sure that the propulsion on the boat is working. Brendan's sitting here in the office. Maritz, who I understand is no longer here, and of course doing a bunch of sampling both with them, but also this incredible Lakes 380 team led by Susie Wood and Marcus. We had the opportunity to get out to the Chathams and do a campaign out there, campaigns here in the Waikato, also down on the South Island, and I'll share some videos of this later on if we have time. And so this is Celeste, who also worked here for a short time at the university, and she was a wonderful technician that traveled with me on many of these adventures. And this is Susie Wood here and Marcus as we're standing on the shore of one of these incredible necklace lakes on Chatham Islands, a dune lake with the ocean just on the other side of that bluff. Of course, I also had the opportunity to engage with a number of iwi, and Brendan took me over to Lake Rotorua, and this is Fia Mary actually using a little cyanoflora instrument. Susie Wood did just an incredible outreach program with local communities. One was at the lake here in Hamilton, teaching students, of course, about lake ecology, but also here's the cyanoflora. Once again, these students were collecting lake samples. And Susie developed this beautiful sort of red light, green light, yellow light system as it pertains to cyanobacterial blooms and lake health. And so being part of these efforts and being inspired by this community engagement was a big part, of course, of my time here in New Zealand. And of course, we made friends for life. This is our Whānau, Vicky, Dave, and the kids. We had many adventures with them throughout our time here in New Zealand. And I just had dinner with them the other night. And of course, everybody's grown up quite a bit. So that's quite a bit of fun. So often when you think you're at the end of something, you're at the beginning of something else. That was the late Fred Rogers. And it is so true. I'm here in New Zealand because of a new project, a new MBIU project, actually funded to study essentially aerial invaders that can come into New Zealand via long distance transport pathways. This project's led by Elsie Pretorius. We're having our first team workshop meeting down on the South Island next week. And so it was originally slated for the North Island in Rotorua, actually, but then it got bumped to Christchurch. So I decided to come here to the North Island to visit with our whānau. Of course, asked Dennis if there was an opportunity to come here and talk and give a seminar, and he said yes. And so I'm very excited to tell you a little bit more about this project in the context of our work, but this is what's brought me back to New Zealand, and I'll be coming back for the next 5 years as part of this project. What I'm really here to talk about is interfaces, and this will be a common theme throughout my talk today. Interfaces are essentially where spaces where different systems come together and interact. And the story starts here as we think about a plant and the atmosphere in an air-plant interface. And of course, here's a dandelion, and as the wind sweeps across the surface of the dandelion, of course, you have different systems, different things happening, a lot of really interesting biophysical systems as well, from the wind detaching these seeds to the seeds being transported in the wind, of course, to new locations. Of course, we have the air-water interface as well, and emblematic of this is the beautiful kingfisher as it's diving through the air, down into the water, going through that air into that water interface, of course, grabbing a little meal. And now the challenging part is getting back out of the water to the air. And so the processes and systems I'll be describing today will be discussing the systems in place at these interfaces, in particular the air-plant interface and also the air-water interface. And of course, by studying these spaces, these interfaces, we can learn about the systems themselves and how they're interacting with one another. And this is, of course, a diversity at the helm here, if you will. And so today I'll tell you stories that take us to the air-plant interface. My background and my training is actually plant pathology. I've moved in to water systems largely because of my interest in cyanobacterial plumes and potential for aerosolization and transport of those toxins. And that project, that interest actually is what brought me to New Zealand as part of my Fulbright. But my training's in plant pathology, the study of plant diseases. And so I'll tell you some of that foundational work and some surprising new mechanisms of spore release and transport. And then I'll tell you about our work, of course, at the air-water interface, deploying all sorts of different robots around the world, including here in New Zealand. Quite a bit of work also over in Europe, as well as in the US. So, of course, as many of you are acutely aware of as a result of the COVID-19 pandemic, the atmosphere is teeming with microscopic life forms. We, of course, can appreciate that in terms of airborne viruses, but most people don't think about plant pathogens as being transported through the atmosphere, but many of them are. Of course, you have one cruising around here in New Zealand, myrtle rust, that many of you have probably heard about. That is, of course, transported through the atmosphere. And I'll be talking about rusts today, one rust in particular called wheat leaf rust, which has some interesting parallels to myrtle rust. And so you can be thinking about a little bit about that when I'm talking about that system. This is a field of potatoes that's been decimated by a fungus-like organism, an oomycete, called Phytophthora infestans, which actually caused the Irish potato famine, for those of you who are familiar with that episode in history. And of course, we do these experiments out at our research farm where we grow a healthy crop, we kill it with a pathogen, and then we study how that pathogen can be transported through the atmosphere over long distances. And of course, this requires teams of biologists and engineers working together as part of this effort. And I'd like to formally introduce my colleague, Dr. Shane Ross. He and I have been working together for nearly 2 decades now, studying this process of atmospheric transport. We had an annual review that highlights the sort of concepts, what we like to call highways in the sky, as pathogens can start moving from a source, maybe an infested crop or residue, move through the atmosphere, of course, encounter all sorts of things like UV light, washed out via rain, ultimately landing at some unknown destination for the pathogen. Hopefully it's a new host, a susceptible host, and then that cycle can continue. And in this annual review, we highlight the different scales of transport, and this will be a focus of basically my talk today, the first half, as we consider these interfaces. The pathogen scale literally being millimeters, moving to the scale of the farm, where you're looking at maybe acres, to the scale of course, regions, which in the United States is emblematic of states. We have state boundaries, state territories that regulate movement around the US, for example, at least on the ground, but not in the air. And then we have the continental scale. And I'll put all of these scales within a framework, including New Zealand, during my presentation today. So we'll start with the pathogen scale. Again, this is a scale of millimeters. And as we think about pathogens like fungi, we think about spore-producing structures in particular that can produce, of course, infectious spores that can move from one location to the next. Enter wheat leaf rust. And so this is a fungus called Puccinia triticina. It causes these beautiful lesions, these pustules. They look like little mini blisters, and they're filled with spores. These spores are called Yersinia spores, and these are reinfecting, so they can actually reinfect the host. So from wheat back to wheat. And of course, as these pustules rupture, these spores can be transported through the atmosphere via rain splash, among other things. We embarked on a study to try to understand actually how these spores were being transported via rain splash or rainfall using a high-speed video camera. This is one of my collaborators, Sonny Young at Cornell University. This video camera allows us to record 1 million frames in a single second. So for those of you that played around with slow-mo on your iPhone, your smartphone, you can get maybe 200, 300 frames in a single second. Imagine taking that up to 1 million frames in a single second. You can start to observe some really interesting phenomena, and I'll show you some of those videos shortly. This is a raindrop generator, and so here a little droplet will fall out and it will target basically a leaf surface bearing a pathogen, and then we'll be able to visualize the transport of those spores with that high-speed video. This is Jonathan Barreto, a mechanical engineer. You'll hear more about him shortly, and Hope Grishevsky, really the brains behind the operation. She's a technician that's been with me for almost 15 years, working in various aspects of our plant pathology systems. So our former graduate student, Hyung-Young Park, Dr. Park did some really elegant work. Here you're looking at a surface of a wheat leaf, okay? And the little brown, rusty looking color things are those Uridinea, those pustules bearing spores. You're gonna see a droplet fall right down to the center of the frame. And this video will play a couple times. It's pretty cool. The droplet's gonna fall. It's gonna impact the surface of that wheat leaf, and you're going to see satellite droplets. That droplet will burst into a bunch of little droplets, and you'll see the spores. The spores will be on the outside of the droplet. Okay. Here it comes, boom. Okay, you see the splashing all out on the side. And of course, you can see at the center, look at these droplets, you can see the spores, okay? They're hydrophobic, they're on the outside of the droplet. But something that was quite surprising when we started doing these videos was that the vast majority of the spores weren't actually moving with the droplets at all. Look at this. See all these spores that are being tossed up here actually as part of the impact? Well, as it turns out, when we started monitoring the trajectories of these spores that were being dry dispersed, not with the raindrops, but as a result of the raindrop moving through the air and impacting the surface, we saw these dry spores were being caught up in these turbulent air currents. And the vast majority of the spores were actually moving via dry air, independent of the droplets, which was counterintuitive and something unexpected, and ultimately resulted in this beautiful paper in PNAS that described these microscopic vortices. And so essentially, as a raindrop impacted the surface, these vortices were generated. This is a PIV model essentially showing each trace of each spore as it forms a mini tornado, carrying those spores up into the air and over some distance away from the leaf. This right here on the x-axis shows the droplet velocity and then the number of spores. So as the raindrop velocity increases onto the surface of the leaf, we see more spores actually becoming airborne, as one might expect. Faster the moving droplet, the more is going to come up. And the same is true when it relates to dispersal distance. This gray line that you see here is actually a model not incorporating those dry dispersed spores. Only those are being dispersed via the water droplets. But when we include, of course, the dry dispersal, we see the vast majority of the inoculum falls way out here and is transported over greater distances, which is pretty exciting. Now, Jonathan Barreco is an expert in, well, he's a mechanical engineer, but he's an expert in surface chemistry and also condensation. And so he's been working a lot in this space of condensation and dumping droplets. So when condensate actually forms on the surface on a hydrophobic surface, when the droplets of condensate actually collide, a really interesting phenomenon happens, and that is the droplets can jump away from the surface. And so as these droplets come together, the energy of the collision is transferred and they can spontaneously jump away. And so we hypothesized that, hey, jumping droplet condensation might actually be occurring as a fundamental mechanism for pathogen transport. So to look at this, we set up a high-speed video camera. We, of course, created conditions that allowed condensate to form on these wheat leaves. And former graduate student Dr. Nath observed, of course, these jumping droplets as they moved away from the surface. You can see them there. So once again, droplets are coming together, they're colliding and then spontaneously jumping away from the surface. And you can see that in these different panes, and we'll look at it in a variety of different ways. Sometimes the droplet jumps, it lands, it collides with another droplet, and then it jumps again, almost like pool cues, for those of you that like to play pool, as these droplets jump around. You'll see that here. There's a trace. The droplet is actually going, landing, and then boom, it bounces again as it collides with another droplet. Okay, so can these droplets really be carrying spores? Well, this is the money shot. So you're going to see from the top down now, looking at that infected leaf, you'll see a droplet actually jump towards the camera. So look here, you'll see a droplet, boom, jump away from the surface. Now, what's even more impressive about this is that, of course, spores will be carried in that droplet. Now, you'll not only see it in the video because there are 3 Uridinius spores, you'll see them actually be carried away with that droplet as they jump away from the surface. But we found a way to actually quantify the number of spores in the droplets using of course, the spores are now gone, had to put that into the video, in terms of dispersal distances and mechanisms. And Jonathan really liked the idea that these plants were sneezing. And so our first paper, he actually put the sexy title, Sneezing Plants, Pathogen Transport by Jumping Droplet Condensation. The idea that the plant is trying to expel, of course, this agent that's on its surface, of course, personifying what's happening here with this phenomenon. And so another graduate student, Dr. Mukherjee, looked at the synergistic effects of the jumping droplet condensation and also wind. This was subject of another PNAS paper where we took an infected wheat surface, we generated the conditions favorable for condensation, and then we had a little fan. We could adjust essentially the speed of the fan to then blow across the surface. These are water sensitive papers. They're yellow, and when you get a little bit of water on them, they actually turn blue. And so when a droplet lands on the surface of those water sensitive papers, of course it turns blue, and then you can spot the droplets, and then of course spot the spores inside of them. So this is where 2 droplets landed on one of those papers, and then you can physically actually just count the Uridine spores that travel in the droplets. And so Dr. Mukherjee did this, looking of course at different wind speeds. Here, 0.5 meter per second, and on the bottom, 1.5 meters per second. Different distances on the x-axis, which represent different distances away downwind from the source, and then the number of spores. And of course, as wind speed cranks up, as one might expect, the number of spores dispersed at greater distances from the spore source increase. And so there is definitely a synergistic effect of both wind and jumping droplet condensation as part of this phenomenon. We'll move now to the farm scale, looking, of course, at tens to hundreds of meters, the crop field, infested plants or residues. And this story takes us back not only to that Irish potato famine I mentioned, a big biosecurity project, which was part of my first major grant at Virginia Tech, to study the long distance transport of Phytophthora infestans from infected fields. This is what the organism Phytophthora infestans actually does. It basically kills the plant host, here a potato, and then it creates these halos of sporangia. These sporangia are on sporangiophores. They're like microscopic lemons. They can pop off, move through the atmosphere, and if the conditions are right, when it lands, that little lemon can actually turn into 8 swimming spores. So you've got a pathogen here that can fly and that can swim. And this was this is in large part, of course, also the lack of host resistance, which ultimately led to the famine years, wiping out potatoes across all of Ireland. So we set up some experiments where we created a big source of Phytophthora infestans, just like you saw in the picture, and we deployed what is now considered to be sort of an old historical technology. These are remote-controlled aircraft that were, at the time, integrated with sophisticated autopilots. These had Petri plates on the wings, so they open and close like a clamshell from the ground. So we could fly these up, we could open up these Petri plates, and then we could capture pathogens in the atmosphere at different altitudes. And the autonomy portion was something that was particularly novel for us so that they could communicate with each other during all this operation. So this takes us back more than 15 years when we were doing this work, actually nearly 2 decades. In a little grass field, which is now a paved runway. Our drone infrastructure has come a long way at Virginia Tech, but our humble beginnings. I always like to show some of our good takeoff and landings. The unplanned landings you won't see, although you may hear about them. These are 2 aircraft that had what was sophisticated at the time, mind you, state-of-the-art technologies allowing them to speed up, slow down, and basically enter, sample, and exit a spore plume at the very same time. And this enabled us to essentially accurately quantify the number of spores, or sporangia rather, that were coming from the source. And so this advanced our ability to track, of course, a pathogen hundreds of meters from a source field. And this is a result of some of our modeling work, where on the x-axis, we have the concentration of actual sporangia that we observed here within the framework of 100 meters from our source. And then, of course, to our model, which we developed as a result, and a pretty darn good fit for what our model was able to predict was in the air and what our drones were actually capturing. We, of course, have extended this to an entirely different framework now in the context of the movement of genetically modified organisms. So in the US, transgenic crops are, of course, commonplace. And, of course, we're able to set up some very unique experiments, given, of course, certain permissions and permits being obtained. And so we have done some work in Tennessee in a special permitted location. This is a forest that's been clear-cut in the middle of the Appalachian Mountains in Tennessee. And what we have here are 2 plots of transgenic crops expressing orange fluorescent protein. Here, ubiquitous. So that means this crop is producing this orange fluorescent protein in all of its tissues. And then here, only in the pollen. We're, of course, interested in studying the movement of pollen over long distances, with important implications for an individual who's, say, growing a non-transgenic crop that's growing something immediately adjacent to an individual that is. The pollen then, of course, can move and, of course, fertilize the adjacent crop. And so as part of this experiment, we set up traps essentially for this pollen at different distances around the source field, and then also a drone equipped with a novel sensing package, which I'll describe here in just a minute. This will take you through that campaign, show you sort of how we operate. Here's our switchgrass field, the panicles, of course, releasing pollen during discrete intervals during the day. We have a big, this is called MARV, our Mobile Aerial Research Vehicle for transport operations, sophisticated weather equipment, and give you an idea of basically our field site and operations. We have sensors on the ground that are both passive and active. Those that just collect any pollen that comes by just by wind, active, actually pumping, of course, air through the device like a vacuum to be able to trap the pollen. We have sampling arrays set up with different types of equipment at predominantly downwind distances to enable us to accurately quantify the movement of that pollen, both from, of course, an empirical framework, but then also ultimately to derive some of the modeling components, which will be pushing our ability to understand the transport of this pollen over long distances. Of course, when we set up these field experiments, we have a lot of people involved. Our teams are generally a dozen or more on the ground to be able to do these experiments and intensively sample across an interval. Now, the stuff on the ground ends up being, of course, fixed sampling devices. We've developed a lot of 3D printed sampling packages, including impingers, which you'll see highlighted. These impingers take air samples, they pump them through a liquid. The particles are then trapped in that liquid, and then we're able to, of course, quantify them back in the laboratory. And so you might have seen a drone popping up there. That drone will be featured here in just a minute. This is that drone carrying that impinging package, meteorological sensor package as well. And so this, of course, can move different distances, different altitudes from the source, depending on the way the wind is going. And you'll get a chance to see the bubbling going on here right in that impinger, right on front. That impinger can be built for about $1. It's just a 3D printed top built around a 15 mil conical tube. And so having these sort of additive manufacturing technologies that anybody around the world with a 3D printer can, of course, use and adapt becomes very important. And a very similar style of sampling was deployed here in New Zealand as we developed a 3D printed water sampler around a 50 mil conical tube that could be printed by anyone as well. And this is a rather large platform, as you might be able to tell as I'm landing it here. It's quite big with really big propellers that are somewhat dangerous, and so you want to keep your distance. This just shows some of that work in October of 2020. This shows some of the data showing our movement of our transgenic pollen here within the framework of 25-meter distance from our source. Of course, the pollen very close to the source is released in fairly large amounts. It decreases with distance, similar to what we saw within that millimeter scale transport as well. These represent different intervals during the day, with the pollen really being released mostly in the later part of the afternoon. There's the panicles open up basically after lunch for a couple hours, release most of their pollen, and then they're done until the next day, which is really quite interesting and something we were able to observe. Now, you can't actually physically see the pollen in the air. And this is a problem when you're trying to model agents and their transport in the atmosphere. You can't see them. You can't see pollen. You can't see pathogens. But what you can see is smoke, particularly colored smoke. And so we took advantage of a relatively new technology that's been available, and these are called smoke emitters. Now, these smoke emitters really were designed largely for baby gender reveals. And so blue and pink are the most common colors that you can, of course, get. We played with a variety of different colors in trying to do some of our sensing. Now, of course, these aren't true colors as the way it's being implemented on the screen, but you can see a yellow emitter. The emitter, these are fairly inexpensive. They're about 20 US dollars, they're about 35 New Zealand dollars. They last for about a minute and a half. And then, of course, you can do all sorts of wonderful things with these smoke plumes. Here's a blue one, of course, set off. We were tuning some of our cameras to be able to accurately not only image the plume, but we were collecting samples of the colored smoke and then quantifying it with the spectrophotometer back in the lab so that we could compare physical sample collection also with our imaging. And so this just shows some of that work there. This is our now advanced drone facility, which was grass before, but we got money to pave it. If you build it, it will come. Virginia Tech became one of the 6 FAA designated drone test sites in the nation, and this was a large part of that. Here's that very same drone equipped now with an optical particle counter that's giving us real-time measurements every second of particle concentrations. And of course, we can also use that particle counter to distinguish the colors as well. And this just shows how the plumes can change, move with the wind, move with turbulence, among other things. And I'll illustrate some of that in some of our model dispersion in just a little bit. Here, in the absence of wind, of course, the plumes are going vertically. And once again, you can't see pathogens, pollen, these other little microscopic things, but this is an excellent tool for you to be able to visualize where something is going. And in fact, we incorporated these smoke plumes into our future experiments as well, involving pollen, so that we could actually see where the pollen was likely to go and then position our drone basically in the smoke plume. And so this shows some of our modeling framework showing an over top view of a high wind condition with the plume. And so you can see that from a horizontal perspective, when you have high winds, your plume is very shallow, okay? But of course, when the winds are quite low, you get a plume that's much more steep and vertical orientation. And of course, the model is able to pick that up. So if you have real-time inputs from your weather stations in particular, telling you about wind gusts, wind speed, you have an idea of temperature and relative humidity, you can actually then be able to validate these models with these beautiful colored smoke emitting instrumentation. We'll move on quickly to the regional scale. Here, we're looking at something that comprises something like maybe a region here in New Zealand and the US, a state scale, tens of kilometers, large farmland, et cetera. We did some work down in the plains of Colorado, a beautiful state in the United States, where we deployed a very interesting wind sensing package on board our drone. And we tested it against some ground-based instrumentation. And so here's a drone over here flying with a sonic anemometer on top of it. And we have that very same sonic anemometer on a ground station, along with a 3D sonic anemometer right here as well. And then we also flew it adjacent to a tower with a fixed instrumentation. And here's some validation of the drone-based sensor against some of these ground-based measurements. Here, the UAS is this trace, which is in yellow. And you can see it almost perfectly traces, of course, the tower-based measurements at 4 and 2 meters over a sampling mission. Former graduate student Dr. Nolan did this beautiful work where we, of course, took our instrumented package, once again, open source. GitHub files are out there. Anybody can build, of course, the sensor package and integrate it into a drone, mount it onto the airframe. The engineering model is quite simple. You have little bits and pieces that you can print, put all together with a little microcontroller and, of course, your sensor. And you can turn your drone into a weather station. And that's precisely what we did. So it's not just about trapping things, but also recording weather. We did calibration flights over this first little tower system here, which was quite small, near the ground, our sensor on the drone on the tower, and then, of course, flying adjacent to this big central tower, which ended up being home for all these drone swarms. And so we had 150 some odd people out there for this campaign, flying all sorts of different drones, comparing their measurements against this ground-based station. So then we put 2 drones in the air at the same time, and we triangulated sensing between the 2 drones, and then also basically what was happening on that ground-based station. And Dr. Nolan was able to monitor these wind fields across the entire valley that we were sampling, and then also identify using this WARP LES model, Lagrangian coherent structures that were responsible essentially for the movement of a front across the sampling location, largely because we're able to triangulate these measurements. And I'll talk a little bit more about Lagrangian coherent structures as I talk about continental scale, because this phases up right to New Zealand as we consider the continental scale problem. And many of you probably remember, if you were here in New Zealand and late 2019, early 2020, bushfires in Australia, quite dramatic. Now, you may not remember them from the standpoint of if you weren't in Australia, if you were here in New Zealand, you encountered some of that smoke. In fact, on the 2nd of January, so mind you, we arrived in New Zealand in Christchurch on the 1st of January. 2nd of January, we were already on our first adventure. We went out to Akaroa, and my family and I were hiking around Akaroa, and the air quality was horrible because of the smoke that had traveled in from Australia, of course, to New Zealand. And of course, you can see the haze here over the Akaroa area. And then here behind us, this is all haze from the smoke. Our eyes were itching and burning, and the air quality was pretty poor as a result of that. We'll enter Ilse Pretorius. Ilse approached my colleague Shane Ross and I, and she was quite interested in thinking about air bridges that could connect Australia to New Zealand. And so she used the smoke coming from the Australian brush fires as a means to study these potential air bridges. And so here on the top, you can see satellite images of that smoke actually leaving Australia and, of course, arriving to New Zealand over different dates in January. 1st of January, 2nd January, 3rd of January, and the 6th of January. And then these air bridges, which were computed using this modeling framework based on Lagrangian coherent structures. And my colleague Shane Ross really engendered this language of Lagrangian coherent structures in the context of pathogen transport. And so He also went ahead and applied this to potential forecasting for invasions into New Zealand. Here, forecasted at the time, or for fall armyworm. Basically, if it was going to come to New Zealand, where would it be coming from Australia? And so she started looking forward in time along different pressure surfaces. Here, 950, 850 and 700, which correspond to different altitudes, of course, at different times. Identifying these connections, these bridges in the atmosphere by which an intruder or an invader could travel from Australia to New Zealand. This ultimately became, of course, the framework for the successful MBIE bid, which is part of the project ultimately that brought me back here to New Zealand. And so we had a really nice paper, of course, published in Ecological Applications that highlights this work and was the preliminary data that also needed to be able to land that grant. And so we'll be talking about this next week down in Christchurch. And of course, thinking about not only these air bridges, but where do you sample in New Zealand, given this knowledge of these air bridges and their occurrence and their frequency, to be able to set up a framework that will most accurately represent where you should actually be positioned to actually collect something. Okay, so enough about air and plants. Let's talk a little bit about the air-water interface. And for those of you that, of course, are engaged in work in non-terrestrial environments, but into water, this will be of interest. So we moved into this space largely because we were obsessed with this question: how are microbes actually transported from the water to the air? Of course, if you spend any time along the coast, you're certainly aware that breaking waves can do a pretty good job at aerosolizing things. If you spent time by waterfalls, you might have gotten misted, of course, by the aerosols generated by the waterfall. And of course, all of that hydropower also is a pretty significant generator of aerosols. And in the US, we certainly have a number of hydroelectric plants, particularly near my home base in Blacksburg, Virginia. The Claytor Lake Dam produces a considerable source of energy. And of course, here in New Zealand, you certainly have a tremendous amount of hydroelectric power as well. Well, harmful algal blooms caused mostly by toxic cyanobacteria can be problematic when we think about aerosol production and generation. And there's a considerable amount of work that's been done with red tides, for example, where toxins produced by this dinoflagellate called brevetoxins can move inland and cause all sorts of respiratory issues with inland communities. And so we've been obsessed in thinking about the transport of the toxins and associated cyanobacterial cells as they move from the water into the land. Applying a similar sort of framework and scaling it up, one of the first things that we did was actually observe these phenomena inside a controlled flume scenario. Former graduate student Dr. Peach set up a really skinny flume, basically filled it with a solution of bacteria, and then using a high-speed video camera was able to actually study aerosolization processes at different wind speeds. And so the wind comes in here, basically is accelerated across the surface of the water. It then departs the flume, and then the camera is viewing what's happening right here. And of course, when you do this, you can see all sorts of beautiful aerosolization phenomena. This is a very similar technology that we highlighted, of course, with the splashing spores. Here, creating different types of droplets, jet droplets, among other things that are results of these collisions. Of course, when you observe these different phenomena, you can see bubble bursting phenomena as they evolve and particles are entrained and then ejected, including bacterial or cyanobacterial cells. You can see fragmentation where droplets will shear off, taking bits and pieces of material with them into the atmosphere and transported over some distance. Now, the size of the droplet and the nature of that droplet can ultimately help determine some of those transport distances, among other things. And of course, when you apply some rather simple models on probability distributions, you can see how these things shift in terms of bubble bursting versus fragmentation at different wind speeds. Here on the left, 3.5 meters per second, on the right at 4. These shift in terms of your droplet diameter and the probability distributions. And so this ultimately is something that you have to feed into a model if you're trying to understand the potential downwind transport of an agent given certain types of environmental conditions. And so this is exactly what we've been building in and thinking about in the terms of surface scum and large lakes that can be sources of cyanobacterial blooms and adjacent communities that can be breathing air, of course, that could potentially be toxic. Now, we spent a lot of time training our robots to actually study these phenomena. And so I'll show you some of our work out at Claytor Lake, which is about 30 minutes from Blacksburg, Virginia. We've used fluorescent dyes that we've released into the water. This is fluorescent. And we have a robot here with a fluorometer on it. It's actually able to then characterize the plume in real time as it traverses the fluorescent plume here. The sensor's on the front of this robot. And of course, we can coordinate our drone-based measurements also with our robotic measurements to provide not only predictions from the drone images, but the in situ measurements in real time that the robot is making here too. So this is illustrating the concept of coordinated robotics, where you've got an aerial vehicle doing some imaging, and then you've got a robot doing the in situ sampling and characterization as well. This doesn't happen just in lakes, but also happens out in the open ocean. We had a big NSF hazards project where we dumped $50,000 of rhodamine dye off the back of this research vessel, and then we tracked it for the better part of a week and a half out in the middle of the Atlantic Ocean. This happened south of Martha's Vineyard. For those of you familiar with a little bit of U.S. topography, this research vessel, of course, deployed a variety of different floating systems that gave us position and current information, both at the top, but then also subsurface measurements of about 30 meters down at the bottom of the ocean. Here you're going to see this plume of rhodamine dye that comes off the ship. Now, the ship captain was trying to create a spiral, but he did not plan on currents. And he didn't believe me that he didn't create a spiral. He actually messed up the pattern until I showed him the drone measurements and the drone images. And you can see that here. His actual trace was a nice little spiral with no current. But of course, what happened was this large swath of dye. We sent a rib out, made some measurements, of course, of the rhodamine dye at or near the surface, and characterizing the plume within the first few square miles, really within the first couple hours of release. And then, of course, the plume had spread so far, we then had to use the ship to make those measurements. In that early part of the characterization, here, just within a few hours, we were already over a square mile away from that little ship. Of course, the drone-based measurements show that. Here we deployed a drone-based water sampling system, which I'll feature and highlight later on. This is a 3D printed water sampler that, of course, has a 15 mil conical tube. You can swap those out and go out, take images of the dye, and then swing the sample back, swap it out, grab another one. And then we have a fluorometer on board the vessel to be able to characterize that. We have an underwater robot that I'm piloting here also with a fluorometer. So you have surface measurements, and then, of course, the robot can dive down and collect samples. It has a water sampler on the end of it, and then also dye-based measurements. For those of you that spend time on research vessels, they, of course, are built for individuals of much shorter stature than I am. I'm 6'5", and so I hit my head a lot. So I was walking around this ship, the Connecticut. Of course, we spend a lot of time at lakes that need rehabilitation. This is Grand Lake St. Mary's, a hyper-eutrophic lake in Ohio, very close to one of our Great Lakes, Lake Erie. This lake is quite shallow. In fact, its deepest part in the lake is 2 meters. And so I could almost walk across it with my head above water if I wanted to. Not quite, but this also is the source of drinking water for a town of about 3,000 people. And the person who runs the water treatment plant, which has to hit it with about 8 different treatments, is the most popular person in town, because if they do their job right, everybody gets water. Unfortunately, throughout the entire year, levels of microcystin produced by planktothrix, which predominates in this lake, are well above contact advisory thresholds and drinking water advisory thresholds for most of the year, with the exception of the wintertime when you do get some lysing of the cells. So here you have winter, spring, summer, and fall. And this is a decade worth of measurements at Grand Lake St. Mary's. And so it's got microcystin pretty much in it all year round. We deployed some drone-based sampling methodologies. Of course, this is prior to the pandemic. We then came here to New Zealand, and I deployed that similar technology studying lakes across this beautiful country, but I'll introduce this now. The drone has a little simple tether on it, that conical tube, a 15 mil sterile tube, and then on top you've got this little swinging bucket type design that then is connected to a string. And this allows you to collect transects away from the shore, with the idea that the drone goes out, and we deploy this in 10-meter transects. 10 meter, collect a sample, come back. 20 meters, collect a sample, come back. 30 meters, and you can get 10 samples in a control in about 10 minutes. Dennis actually went out with me, and we collected some samples here at Naroto using this very same method. And of course, you then can analyze these samples, and you can see trends here, for example, with sort of peaks and valleys as it associates with different types of congeners or flavors of microsystems. And so you don't see oftentimes this homogeneous distribution of these toxins at surface level. You do see some hotspots, which of course has very important implications for sampling methodologies, by which you would be thinking about whether you want to grab one sample or maybe 10 or more to characterize a lake of interest at a given time. We also did some work, of course, with wind sensing to be able to determine the potential movement of the cyanotoxin, the microcystin, inland. And this is a former graduate student, Dr. Javier Gonzalez Rocha, who's now an assistant professor at the University of California, Santa Cruz, where the drone itself becomes your wind sensor. And so this is a very, very powerful tool in that context. We also did quite a bit of work trapping cyanobacterial cells and toxins inside these small tubes, and we reported that in a recent environmental science paper just this last year. So that very same sampling method, of course, for sampling pollen, for sampling microbes, can also be deployed in this context. Now, where does this go in terms of New Zealand? Well, of course, when I was here, I was taking advantage of opportunities to get out and study lakes that were in need of rehabilitation. Lake Waikare is certainly one of those lakes. And it just so happened, though the color scheme here does not really show it, when we first collected that very first sample shortly before lockdown, 18th of March 2020, the lake was very green. It then had throttled, of course, to an orange, sort of chocolatey color, largely due to monoraphidium sort of taking over. Moretsu used to be here, actually was featured in an article that rolled the October 2020. And I had the opportunity to make multiple visits to this lake and collect repeated measurements. This is Warwick, who used to be a technician here at the university and spent quite a bit of time out in the field. Believe it or not, he's not actually staging this shot. He was down on one knee just helping manage some of those samples. And so we're adjacent during one of these. We deployed the drone, we collected samples, and then we also, of course, deployed a drone with a multispectral camera system that allowed us to generate some inverse NDVI measurements that you can see here related to sort of this color or this throttling of change. So we made 5 different visits to Waikare, which was pretty cool, from the 20th of March or the 18th of March 2020 to September, and then it throttled back to green again, and then sort of sustained itself even into March of the following year with that sort of chocolatey brown color, again predominating with monoraphidium. And of course, we took measurements of phycocyanin and chlorophyll along these transects, and we're able to document some of these trends that were associated, of course, with these shifts in blue-green algae that were occurring in the lake. And thanks to Dennis's great work with this Hidden Gems of Waikato book, he invited us to contribute a feature box, which is part of this chapter led by Ian on plankton, where we describe this technology and just a couple of the sampling dates at Lake Waikare, which is fantastic, and we thank you for that, Dennis. Really proud to be part of that. I did want to mention that, of course, some of our work really extended to some incredible places. If you've never made it to the Chathams, I encourage you to go. It's one of the more unique places I've ever been on this planet. I thought I'd just share with you one brief video here that highlights some of that wonderful work at the Chathams, and you can understand a little bit about how Lakes 380 operates as well. And so this is a series of, I put together these sort of action films, if you will, from some of our different adventures. This is Patariki, a very unique lake, a peat lake that actually is separated by a sandbar from the ocean. You'll see that in a visual shot here. We did drone sampling here on this lake. And some of these images will, of course, feature, that's Celeste down there with some of our drone sampling. This is that sandbar here, the ocean, and then this really unique, almost perfect sort of circular shape. In fact, some garnets were in the sand. If you kind of crawled around on your hands and knees, you could find the stones. This had a little certain unique surface bloom here that we were studying, as well as another lake. And then you'll see shortly some of the Lakes 380 crew actually out collecting cores from these lakes. So depending on where we were at and what we were doing, oftentimes we were sampling at the same time, or we were sampling at different times, depending on our target and what was going on. We linked up with Tom, who actually, his whakapapa goes all the way back to the original Moriori, who, of course, colonized Chatham Islands, and he let us sample his lake on his land, Rotoika. And so this is a Lakes 380 team out there deploying of course, a core, collecting sediment cores at the deepest part of the lake. illustrating some of that. And so they would go out, collect the sediment core. There was another team that went out and collected all sorts of different samples as part of water quality measurements. Of course, there were cattle running around as well amongst these activities. And then, of course, these beautiful sort of volcanic cones that are remnants in the back as well, and some of that multispectral imagery, just highlighting the really incredible topography of the Chathams. This is Susie off here, and Marcus and Tom down here as well. This lake actually didn't have a name, but there was a creek that went into it, and the creek was called Tongaia, so the lake became sort of known as Tongaia. This one, of course, had quite an interesting color to it. Collected samples out there, and then, of course, you can see the Lakes 380 team. This is Kat. She was collecting samples for some pollen work that she's done. She's a pollenologist. Many of you might know who she is. And then we actually, Susie had led a run. She's, of course, stays fit, is always out. She takes a break from her research and goes off for a run. She went up to the top of this volcanic cone, and then we followed her to get some drone images of the lake, and you'll see that shortly here in just a little bit. And here we are up on the top of this mountain here, and just a cool little pan of the channels. There's music too, but I'll turn that down. We started about 10 minutes late, so I'll just finish with something we're doing right now, and then hopefully there'll be time for some questions. We've been moving a lot of our work into the study of air-ice water interfaces, a sort of a new phase of our work. And so, of course, with climate change comes the collapse of many of our glaciers, and calving phenomena like this one that you see here, of course, causes a tremendous amount of material to enter environments. Some of these calving events have caused increasing nutrient loading in regions, which have led to cyanobacterial blooms, among other things. And so we're interested in studying processes by which cracking and basically melting of these glacial environments can cause particles to move into the water and also into the air. This is a security issue in the US. In fact, we have a lot of people who like to go ice fishing. And so in the Great Lakes, just back in January 2022, a big sheet of ice broke off. People were ice fishing, and of course, they didn't have boats. They'd driven out on vehicles, and so they had to be rescued. And so understanding cracking phenomena extends not only to glaciers, but also to present-day ice fishermen as well. So we've been doing some work in the lab, taking artificial ice that we can make in the lab, seeded with dye, and then we've been watching it basically melt, and then understanding sort of how that meltwater is transported through the water, and then ultimately we'll be doing aerosolization experiments. You can see that meltwater shedding the dye here, and of course the rolling of this ice ball, which is quite interesting. And then we've been using, of course, thermal cameras to examine this phenomena. So here you're going to see the cold sort of in this darker blue, and then the warmer area of the warmer water, of course, is more of a yellow. And you're going to see here a top-down look, and you'll notice this cube basically flip. And what you'll see are tendrils of meltwater that you can observe largely due to this heat signature. So that's pretty cool. So you can't see these with the naked eye, obviously, unless you're looking at actual dye. This is meltwater that's moving throughout here. So using technologies like this, we can begin to understand these melting and cracking phenomena. And that's something that's obsessing our work at the moment. And we just got a new grant funded to actually study this at Lake Mendota, which is near University of Wisconsin-Madison. So we'll be deploying technologies to study these phenomena in the coming years. So with that, I still say that I do have 10 minutes because of our late start. Hopefully there's some time for some questions. Happy to take them if any of you have anything. So appreciate the opportunity. Thank you.
Yeah, people are happy to stay around. Absolutely, we have time for questions. Open the chat box to.
Yeah, go ahead. Yeah, go ahead, Whitney.
It strikes me that a lot of your data is visual, and I'm wondering about like some tools that you use to quantify that, and maybe the balance between like a human observation measurement versus maybe some more machine learning or other technologies that you guys use.
Yeah, I appreciate that. So one of my graduate students who just finished up, he actually defended last week, actually has taken some of our data in the imagery space, particularly with the colored smoke plumes, has applied some machine learning models to actually quantify or make estimates of those concentrations, and then we can calibrate those images and those estimations with our empirical data on the ground. And so we did a little bit of that with our dye work out in the ocean, in the Atlantic Ocean, as part of being able to use the drone images to then predict the concentrations that we actually see with our fluorometer. The same is true with the color smoke measurements, but we haven't submitted that paper yet, although he promised me we're going to get that out hopefully in the coming weeks, but his thesis has been submitted. So it's a great question. So there's a lot more to do in that space, and AI and ML will continue to be part of our future. I don't think we'll ever be replacing empirical measurements, but we'll be able to probably make far fewer empirical measurements and lend sort of these other sort of large scale imagery-based estimates to drive some of what we're doing. So great, it's a great point, especially as we move into satellite imagery, among other things. Great question. Others? Brendan?
Yeah, hey, so great. Thanks, David. So absolutely wonderful. Great stories. I remember some of them. So
You were part of a few of those, Brendan.
I was part of a few of them, but your background is astonishingly similar to mine. I was a plant pathologist.
I know, I remember this, yes.
In a previous lifetime. So I'm conscious that the myrtle rust, and which was, and possibly still is, a serious problem.
Yes.
But it sort of seems to have had more limited effect than we sort of initially thought of. We thought we'd lose all the verticalas and all the sort of Metrosideros type species. So does your work say anything about dispersal as a possible agent for why we haven't a spore dispersal? Or is it possibly more than just a few more resistant plants that we thought we had? Because dispersal or resistance.
So yeah, it's complicated. With the short story is with rust, it's complicated because you need a susceptible host, but you also need conditions favorable for infection, and that includes, of course, a susceptible host. But most importantly, you need water. So rust spores are intimately tied to water. Usually, there's an 8-day infection cycle for most rusts. The spore lands, you have to have water, and then you'll get your first pustule within about 8 days. Okay. In the absence of water, if it's just dry, you can have the spores be produced and blown, but if there's no water, then the spore will only remain viable for so long, and then it won't end up infecting the host. And so that water component is particularly important and probably explains in part the somewhat sporadic nature of the disease, while you'll get hot pockets of myrtle rusts in certain areas, but not in others. In fact, when I entered through Auckland, mind you, a few days ago, of course, the biosecurity personnel asked me what I was doing here. I honestly shared the whole story coming for this biosecurity bit. He proceeded to ask me more. I literally stood there for probably 15 minutes at his window because he was very interested in myrtle rust. And so we had a very similar conversation to the one we're having right now. And he was so excited to be talking to somebody who'd done work with rust. He actually took me away and he walked me around to show me where to go to put my bag into the scanner. Whereas the people ahead of me, he just pointed and sent them on their way. So anyway, a funny welcome to New Zealand. He also asked if I brought any rust spores with me as part of my stay. Of course, the answer was no. So I don't know if that gets at your question, Brendan, but it's a complicated nature of host, of environment, and then of course that atmospheric transport and dispersal event, which is something that we're hoping to get a pretty good understanding on as part of this MBIE project. And it includes insects too, not just pathogens, but my expertise, of course, is in the pathogen arena. Insects have behavior, which creates a whole nother really complicated aspect because they can make choices about where they go and what they do.
The limited viability of the spores had no idea.
Yeah, well, that certainly is an issue. And so one of the criticisms, a lot of folks will do DNA-based tracking of agents in filter samples, among other things, but that won't tell you anything about viability. And so a lot of these sort of baseline collections, which are wonderful, can tell you about these potential invaders. But of course, if it's all dead, then it's not going to really matter in terms of it's going to get established. Yeah.
So you mentioned that pollen and spores tend to not have a visual component when they're dispersed. Do they have a chemical component that could be sensed in lieu of a visual kind of component?
Yeah, I mean, that's a really good question. So Andy Alt at the University of Michigan in the US has been looking at signatures of sort of aerosol signatures that are associated with the transport of other things. And so if you monitor these other things that are associated with, say, a pollen grain or a cyanobacterial cell, then you can use that as a proxy to study the agent of interest. And so I think we're really just in the infancy of that. But I think the short answer is probably in the future, we can look at studying other things to study the target organisms that we want to actually understand, if that makes sense.
Yes.
Yeah, how are you? Great talk, very interesting. I'm just curious, the NDVI kind of imagery you were showing there. What kind of data do you get out of that, and how do you?
Yeah, so what to be honest, we haven't published anything from any of that imagery yet. Our hope was to be able to use some of the imagery to better delineate the spatial distribution of the HABs on the surface. And so the RGB images are somewhat limited in that capacity. And so interestingly enough, we did some work with Brendan out at Lake Ohinaway, and Ohinaway had a really interesting sediment trail where the inlet was basically depositing the stream was coming into the lake, and we saw some really interesting sediment signatures of transport in our NDVI images that we could not see in the RGBs. And so the short answer to your question is there's a tremendous potential to take advantage of the multispectral camera systems. We haven't fully realized yet that, but we have all the data. What I'd really love is to have a couple students or some postdocs mine this treasure trove of digital imagery that we have of these lakes and be able to relate that to the empirical data that we have for the sampling. And then that's kind of where we want to go. And so, in fact, Brendan is part of an Ion Lakes project, which I think has ended, if I'm not mistaken, Brendan, which was really looking at hyperspectral signatures of these different lakes and really developing an extensive data set in that capacity. I think there's taking those type of data, overlaying them with drone-based data, satellite data, there's a tremendous potential to use those signatures to ultimately predict water quality. And it comes back to kind of Whitney's question, can you get to where you can take a remote sensing measurement in any capacity and not have to rely on empirical data? And I think the short answer is we're moving in that direction. Once you can tune your system and understand what an image actually means, what it stands for, then you can turn the technology loose. And we're seeing more of that.
And I'm just curious how this was all the geospatial data is collected with your camera, but I guess working with the images that just kind of like you look here and like.
No. So the beautiful thing about this is everything is geo-referenced, and so some of my students have developed orthomosaics of entire lakes, and so you can literally and it's quite easy to do now. It used to be quite laborious, but it's all automated. And so you take all your images, provided you fly over the entire lake and encapsulate it, you can create a huge orthomosaic of very high resolution across the entire lakes. And so all of the lakes that we studied that were within the right amount of size to cover on one battery of the drone, we have covered the entire lake for that purpose. And so that comes back to that question of what do you do with all the data? So at every lake we sampled, I flew 3 drones. The water sampling drone, the drone to get pictures and movies and images to send off and share with Dennis and Susie and whoever wants to see them. They've been made available for their team to use however they like. And then the multispectral drone. So every lake we appeared to, we always had 3 flights. Yep, 3 up, 3 down, and no casualties at all, which was of people or robots.
We're very grateful for those images.
Yeah, sure. It's I'm pleased to see that they're being used in a variety of capacities. Great.
Awesome. I might just draw the questions to close, but feel free to join us for lunch after that. We'll take David out for lunch and just continue the conversation and the questions there. Please join me in thanking David again for making time to give a seminar here. Thank you. Appreciation, David. This is a small tonga, which just sort of reminds you of that you gave the seminar. Yeah.
Oh, that's fantastic. Thank you, Dennis. That's beautiful. Appreciate it.
Cheers.
Thank you. Yeah, that's beautiful. Thank you. Thank you, everyone. Appreciate the time.